Hi guys, welcome back. Today we're going to finish up our conversation on glycolysis, which is the pathway that generates pyruvate from glucose. Before we get any further into our discussion, I just want to do a brief recap of what we've already talked about. So here on the left in red, we see the glycolysis pathway. Remember, this is the conversion of glucose to pyruvate, and this occurs in two separate phases. The first phase is the preparatory phase, and this sees the glucose be phosphorylated into that glyceraldehyde 3-phosphate. Once we have the glyceraldehyde 3-phosphate, that is then oxidatively converted to the pyruvate molecule through the payoff phase. The most important thing to take note of here is that at the end of the preparatory phase, there are actually two glyceraldehyde 3-phosphate molecules present. This is why you see two pyruvate being generated at the end of this pathway, because then throughout the payoff phase, there are two molecules produced at the end of each individual step. So now that we've just done a basic general review, we'll look at where we left off last time. We briefly discussed this reaction. This is the 9th step in the glycolysis pathway, and it sees the conversion of 2-phosphoglycerate to phosphoenolpyruvate with the use of enolase, and this is a dehydration reaction. Enolase is an important enzyme to note, and you should know that because this reaction is reversible, as indicated by the bidirectional arrows, enolase can be used to catalyze the forward reaction of 2-phosphoglycerate to phosphoenolpyruvate, and it can also be used to catalyze the reverse reaction of phosphoenolpyruvate to 2-phosphoglycerate. I've just included the energy of this reaction at 7.5 kilojoules per mole. Keep in mind that this means because it's positive, it's endergonic and non-spontaneous. So we're finally moving on to the last step of glycolysis. This is where we see the pyruvate molecules actually being generated. And I highlighted here that just to keep in mind, there are actually two molecules of phosphoenolpyruvate, which is why there are two molecules of pyruvate. And because there are two phosphoenolpyruvates, there are also two ADP molecules. And what's important about these ADP molecules is the phosphorylation of ADP to ATP utilizes the cofactor magnesium. I've highlighted magnesium by the unidirectional arrow just to keep in mind that magnesium is the cofactor of this phosphorylation. It helps to release the strain on ATP, and while it's almost never drawn, it is always present, and you guys need to be aware of that. The most important part about this reaction to keep note is that it's an irreversible reaction. So pyruvate kinase can only catalyze the forward reaction of phosphoenolpyruvate to pyruvate. If you wanted to do the reverse reaction, it would involve a separate enzyme and an entirely separate pathway that we will talk about later. But now that the pyruvate is synthesized, we'll talk a little bit more about that molecule specifically. So first, when the pyruvate is originally synthesized, it's in its enol form. The enol form is shown in this image on the left. It has the central carbon double bonded to a CH2. This is energetically unfavorable. It's low energy, meaning that energy then can't be put towards other biological and cellular processes. So to combat this enol pyruvate will undergo the process of tautomerization, and this generates the keto form of the pyruvate. You can tell it's the keto form of the pyruvate because there is a ketose group or a carbon double bonded to an oxygen off the central carbon of this molecule. This is incredibly high energy. It's negative 31.4 kilojoules per mole. So that is why it's so favorable. That energy can then be put towards, as I said before, biological and cellular processes. So while this is a separate step in the glycolysis pathway, it's not written as such. So just keep in mind that originally the pyruvate is synthesized in the enol form and is then tautomerized to the high energy keto form. So that pretty much ends the individual steps of glycolysis. So I just want to emphasize the products here. Pyruvate is obviously the main product that we want to look at, that we focus on. So you have two molecules of pyruvate, but along with that, you also have two NADH, two H+, two molecules of ATP, as well as two water molecules. So now the discussion becomes what happens with that pyruvate. The cell has gone through all this work, used all this energy to make it. Why? And that's what we'll look at now. So pyruvate, once it's generated, can undergo several different pathways depending on the environmental conditions at the time. So if there's anaerobic conditions, which lack oxygen, pyruvate will be fermented, and it can either be fermented to lactate or ethanol. Here we'll look at lactate fermentation. This is a process specific to mammals. So humans, we perform lactate fermentation, and this sees 2-pyruvate being converted into 2 lactate molecules. And this uses the oxidation of the NADH produced earlier in glycolysis back to the NAD+. This is an important process because lactate is used in the brain as a signaling molecule, and it can also be used as a form of energy. But if you're this, as I said, this is specific to mammals. What if you're in plants? In plants and yeast, they'll undergo ethanol fermentation. And ethanol, as you may know, is drinking alcohol. And this is something humans utilize for their own use. So humans will ferment yeast to create alcoholic beverages like beer, wine, and liquor. So that covers what happens to pyruvate under anaerobic conditions. What about aerobic conditions where oxygen is present? If oxygen is present, pyruvate can be synthesized into the molecule acetyl-CoA. You might have heard of this molecule before as it's used in the citric acid cycle, but it has a particular use in the regulation of glycolysis. Acetyl-CoA is associated with high energy, meaning there's lots of ATP. It's a proportional relationship, high acetyl-CoA, high ATP. ATP is another one of those products of glycolysis along with pyruvate. If you have a lot of energy, if you have a lot of ATP, you don't need any more. So what acetyl-CoA does is it actually goes and inhibits the enzyme pyruvate kinase that we just discussed. This inhibition prevents any more pyruvate from being synthesized. And since pyruvate is the main product of the glycolysis pathway, it essentially just inhibits the pathway entirely. And it will be inhibited until the acetyl-CoA levels go down, ATP levels increase, and then the pyruvate kinase is stimulated to produce more. So that is just something interesting that can happen with pyruvate once it is generated. And with that, we have wrapped up our discussion on glycolysis. We've talked about the 10 steps of the pathway, the enzymes used, what happens afterwards. And on the right here, I've included this very important diagram. I think you guys should do your best to commit this to memory. I've included in blue the molecules, what's converted to what. In purple are the enzymes that do the converting. And then in green, I also have the payoff. So it can go from ATP to ADP, the reverse ADP to ATP. I also have included the reduction of NAD+ to NADH. This is an incredibly important image. If you understand this, you understand what the enzymes do, you understand the pathway, and then it can be used to answer any questions about glycolysis further on down the road. So with that, we'll end here today. Thank you guys so much for listening. I'll see you next time.